Okay. Well, buongiorno a tutti, and welcome to today's colloquium. It is my great pleasure to introduce to you Professor Alberto Diaspro. He's a biophysicist, professor of physics at the University of Genova, and director of research in nanoscopy at the Italian Institute of Technology, located also in Genova. In confirmation of the interdisciplinarity of research in biophysics, Alberto graduated in electrical engineering from Genova University. Then he spent a few years as a lab technician, first in the Biophysics Institute and then in the physics department at Genova University, where he was promoted to full professorship in 2018. He is a worldwide expert in optical nanoscopy and nanoscale biophysics. In particular, he is well known as a planner, designer of novel optical methods that can be successfully be used in molecular oncology, neuroscience, and material science. For all this experimental activity, he was awarded numerous prizes. Here I just mentioned a few of them, starting from the Emily Gray Award from the Biophysical Society in 2014 for significant contribution to education biophysics, the Gregorio Weber Award for Excellence in Fluorescence Studies, 2022, and more recently, in 2024, the Enrico Fermi Prize from the Italian Physical Society for his contribution to optical microscopy and his impact on molecular biophysics. In the same year, 2024, he was nominated Cavaliere dell'Ordine al Merito of the Italian Republic by Presidente Mattarella. In today's colloquium, he will show us how modern optical microscopes, with the help of artificial intelligence, can produce details, quantitative details, well beyond the diffraction limit on the molecular mechanisms taking place in living cells. And of course, the very ambitious goal of this research is to create an artificial optical microscope to see what we could not perceive before. So, thank you very much for accepting our invitation, and the floor is yours.
Thank you. Thank you very much for your presentation, your kind presentation. Thank you for being here. It's really a great honor for me having this chance to give a lecture in this room. The title you see here is the artificial microscope, and we will discuss about label-free and fluorescence microscopy at the nanoscale. But since at the very beginning in this presentation, some words were mentioned, I start, the last one is the Fermi Prize, but what I would like to tell you is that I started with these guys in 2003. Maybe you know some of them by name. And so I have to thank them for what we have done in all these years. This is one of the pictures we had for the last group in my lab, including some experts coming, visiting the lab, Samantha Cristoforetti in this case. And here in Padua, there is a person that I learned a lot. And this guy, I don't know if you know this guy, but was Tullio Pozzan. And so I'm really indebted with Tullio Pozzan, myself and all my group, so we share different schools. And in terms of dissemination of science, I think that also here in Padova, you had a great person that was Pietro Greco. I don't know if you know him, but in case you don't, please go to the Bo and start learning or reading something written by Pietro. Now, behind the artificial microscope, there is the idea of observing, watching, and trying to understand. And so I like to start with this sentence by Galilei, And there is a story behind, and this is a story of a student, of a young guy, that decided to bring his telescope around and to try to understand if people, if they were interested in observing something that they couldn't see before, they couldn't see without using this, this instrument. In a comparatively short time, people was attracted and started to watch. Started to come to go to his telescope, observing and having comments. Everybody was attracted by this chance of observing. And one of the objects that was more interesting was the moon. And starting from the moon, we can switch to this picture, and this is a recent image of the moon, and this is what Galilei was able to draw about the moon. When he wrote, and I don't know if you had the chance to read this book, Desiderius Nuncius, is a very short book. It's a real revolution in science. Is a book that Pietro Greco, when discussed about this book, called The Dangerous Idea by Galileo. The dangerous idea are 3 words: observing, explaining, and disseminating what we have seen. And this was really a revolution at that time. And this was something that Pietro was interested in, and I think all of us. And if you have the chance to read this book, you will find this an exceptional book, very short but rich. Now, we will deal with pictures as an exhibition. I would like to say molecules at an exhibition, recalling this opera by Mussorgsky. And it's really intricate, the world of the molecules in the cell and in the living system. And so our story can start with the Emperor of Light. The Emperor of Light, because here Magritte reported about natural light, artificial light, light that can be controlled by humans, light that can be used to see something, but still there is something that is behind, that is unknown. And this is something that can produce some enthusiasm when you watch. This is from the Fabelmans. That is a recent story. But behind this story, there is this one. Someone giving you an instrument to explore what you are interested to explore, to watch something. Doesn't matter where you can have your final image, in your hands or in a screen or everywhere. This is the story of Steve Spielberg. Probably you know about it. It's a story not only about light, but since I'm interested about light, I continue with music and with Pink Floyd that you know, that we are splitting light. And I'm interested in this because I'm interested in white light and in colors and in different wavelengths. Of course, not only in terms of books, of music, but also in terms of science. And since we are here, and here, if we have Galileo in our minds, this sentence tells you that the term microscope was born starting from the occhialino per vedere cose minime by Galilei. And when this instrument was known, was understood in terms of its power, there was a great experiment that the Academy of Lincei, they started. This book is the apiarium. They used an optical instrument to watch bees. But this is what they had in mind, a great experiment that aims to create a method for the scientific ordering of all the knowledge about the world achieved up to then and under construction. But the most important part of this sentence is combining the written and figurative science. Now, figurative science. I mentioned artificial, so learning is something that you can have in your mind. So I would like to remind you that there is a category of studies that are related to visual learning. This is a very recent paper telling about what is behind our vision computationally recreated. It's very interesting. I don't want to explore all these aspects, but only this one that we are interested when we deal with single molecules, navigation, detection, and tracking. But also to tell you that in Italy there was and still is Tommaso Poggio that started working in artificial intelligence using images to train an artificial intelligence system. Now, today it seems that it's something new, brand new is not. It's part of the story of Tommaso Poggio that started this work in Genova when he knew a scientist that was the founder of biophysics, Antonio Borsellino. Antonio Borsellino was a theoretical physicist. Tommaso was a student, and then other generation were students. And the idea is that you are linking vision, neuroscience, understanding what is around, what you can build, what is real or not, all things that today we are trying to explore using more sophisticated systems. And in 2024, you had this Nobel Prize. I think that Tommaso Poggio could be here. Apart from this, but these guys, they use physics to find patterns in information. In the information channel, that is the one we explore using the microscope and using light. However, my hero, my heroes are not the ones I mentioned. My real hero is this guy, is Maxwell. My hero is Maxwell, and the reason is comparatively simple. I will try to convince why, but I think that you can be easily convinced about this. But think about the battery that you can buy in the market. This was a discovery by Alessandro Volta. But when electricity allowed us to have a big jump in technology, when Maxwell wrote the Maxwell equations, transforming these in waves, transforming these in something that could be used, and in the idea that you didn't need any wire to transport information. And here with the optical microscope, we are using only a very small portion of the electromagnetic spectrum. We deal with the speed of light. This is the real time needed in case you are able to shine your photon to the moon. And I'm mentioning this because when I go into the cell, the biological cell, that is one of the objects I'm interested in, the characteristics, physical characteristics of these cells tell me that passing through, for passing through, the time is very, very short. So using time for studying what's going on, even if there are changes in your refractive index, is really a challenge, more than a challenge. And so we have to find different ways. The cell, biological cell. Just reminding you that there was a book, Micrographia, wrote by Robert Hooke. And in the book Micrographia is reported for the very first time the name cell. So the origin of this name is linked to the observation using a microscope. But when we deal with cells, biological cells, the ones we have in our bodies, we have a problem. We are in the region of visible light, and our molecules do not absorb visible light. In case they do, there is an increase of temperature, and so it's difficult to stay alive here. And so in case we want to produce some contrast, we have transparent objects. And so we have to remember that what happens when we have light passing through is that we are able to collect by our eyes the intensity with our detector intensity, and so we mix the phase and amplitude information. Amplitude is the same, phase changes. I don't know if you have a ChatGPT, or if you use it, or if you use any Google research engine or whatever it is. You can try to ask who was the first Italian getting the Nobel Prize, and I can tell you the date, 1906. I don't know if you know the answer by yourselves. Probably, ChatGPT will reply, Giosuè Carducci. That's true. Giosuè Carducci got the Nobel Prize in 1906. But there was also a scientist that got the Nobel Prize in 1906. But since interrogation in ChatGPT are more for the humanistic side of the country, of the world, the answer is Carducci. The other Nobel Prize was for Camillo Golgi. Camillo Golgi is not the one of the organelles in the cell. He's a guy that discovered a way for having visible neurons. Otherwise, they're transparent. They don't absorb light with a black reaction. Years after this, you had the Zernike finding a way for transforming phase changes in intensity changes, introducing phase contrast. So our black and white world started becoming being out from the gray zone, becoming colorful. And today, we are dealing with color images. We have some substances, we will discuss immediately about them, that can assign colors. And we can, using what I call at the end the artificial microscope, the idea is to transform a black and white image in a colorful image, providing molecular content. Colors come from a medium of contrast that are called fluorescent molecules. So the mechanism is the one of fluorescence. And if you ask David Jameson, that was one of the pioneers in fluorescence, why fluorescence, the answer is it's pretty. This is the very first answer. But then it's pretty because fluorescent molecules, the fluorescent probe, can provide information about electric fields, viscosity, polarity, pH, temperature that are around the fluorescent molecule. And you can position your fluorescent molecule in a very specific place in the biological cell. So you can have fluorescent molecules bound to DNA, bound to RNA, bound to other organs. So you can gain in specificity that you don't have when you perform label-free imaging. You are not sure of the origin of the signal. So for example, here, this is a sample that you can find in any park around, in pond of water. It's paramecium. And you can use different colors, different fluorescent molecules emitting at different colors, labeling different components of these biological systems. So you can get specificity. There is something that has no contrast, and now you can find specific components, and you can have them in colors. Mechanism is not, well, it's more complicated than one can think. And we are working, I didn't tell at the very beginning, but I can tell now, and I will tell later, reminding this you later, we are working room temperature, atmospheric pressure. So when we excite a molecule, that is a fluorescent molecule, we have different pathways. We go to an excited state, and from the excited state, we are back to the ground state with emission of photon and having many other mechanisms around. But the main mechanism is this one, and so you are cell loaded. You have the chance of providing energy for exciting the molecule when the molecule is in the excited state. You have a decay, you have a relaxation at different wavelengths or energies. You have a phenomenon that is, you can compare with a radioactive decay, even if temporal window is completely different, but is the fluorescence decay. I bring to your attention this fluorescence decay because we are able to measure this fluorescence decay. What does it mean, having fluorescence decay? Different fluorescence decay means that I bring the molecule to the excited state. It's a fluorescent molecule, so the main activity is emitting photons to relax. But there are also other ways for relaxing, dissipating, or other non-radiative things, or other interaction with other molecules. And so the different ways for relaxing influence the decay of fluorescence. So in case you have a sample and you have a red emission, like in this case, from the red emission you can understand something about morphology, but if you build a map having false colors that report about what is called fluorescence lifetime, you can have different colors as function of the different environment explored by the molecules involved in their relaxation. The different ways they can relax that are related with the environment. There are several ways for bringing the molecules to the excited state. If you need energy 10 blue, you can provide the 10, you go to the excited state and then you go back. Or you can use the two-photon excitation or multi-photon excitation, exploiting the thesis work by Maria Goppermeier and other works by other scientists in the due time. What is relevant here is doesn't matter if you bring your molecule to the excited state using energy 10 or 5 plus 5. When the molecule is in the excited state, relaxation is the same. And so if it's a green emission, it's a green emission in both cases. Since two-photon absorption is not a natural phenomenon, a phenomenon that you can experience in a simple way, you need a very high intensity, a density of photon to promote this event. At the highest density of photon that you have when you are using a microscope is at the focus of the lens, because here area becomes 0 and number of photons is the same. This means that when you deal with conventional excitation, you have a double cone of excitation, so you send your photon across this double cone and all these photons are able to excite the molecule and to get fluorescence back. When you use two-photon, you are highly localized. So you can select regions in your cell, and you can better understand what's going on region by region, in this case, release of some molecules from the inner part to the membrane. These are the very first elements that we have when we develop microscopes or when we use fluorescent microscopes. But also at the time of Robert Hooke wrote this sentence: there is nothing so small as to escape our inquiry, because he had the microscope in his hands. The idea is that something that was not visible was visible due to the microscope. And so this was enough. Now, let's start from this sentence, and the other sentence here, and then you see which is the spatial scale that you want to explore: the human body, human brain, skin, cells, bacteria, protein, amino acids. And you have different tools. You have your eyes, you have conventional microscope, you have super resolved microscopy. And in my case, I'm interested, and today we will end with Ångström fluorescence microscopy. In my case, I'm interested in understanding the mechanisms of condensation and the condensation of chromatin DNA, that is something in the nucleus of the cell. And just to give you a scale, these are 100 nanometer. These are the nuclear pores. This is chromatin, the nuclear lamina, and something that is inside. But what is nice with this sample, apart the scientific interest, is that it's a kind of El Dorado for a microscopist. Because the dream here is to use the very same instrument for exploring what's going on from 1,400 nanometer down to 2 nanometer. This is the chance you have when you have an optical microscope in your hands. And during all these years, every 5 years, more or less, there is a change of view related to the way DNA organizes in this nucleosome, around these nucleosomes, organizing chromatin, and organizing the release of information. Can you imagine? You have a nucleus that is 1 micron, let's say. Inside you have 2 meters of DNA that is compacted, and that, when is needed, it opens a small window, releases some information, and the cell proceeds in its action, and you stay well or not. Now, if you go to the microscope, at the very beginning we have the wide field, so-called optical microscope. We are diffraction limited. Then we started using some other tools, and we will go inside to some of them, for improving spatial resolution. And today we are here, below 10 nanometer. Again, room temperature, atmospheric pressure. And you can use this tool to study different things. You can study the way a cancer cell becomes a cancer cell, why children were born old in this disease that is called progeria, that maybe you know from the movies, or in neuroblastoma, that is a very bad disease for children. And so the idea is to build a microscope having all the possibilities we are able to manage in the very same instrument. It's a kind of orchestra. In this orchestra, if you never were listening these sequenzas by Luciano Berio, in case you have the chance, do that. It's amazing. Because here you have single instruments, or microscopes, or music instruments. They play at the perfection, one by one, but it's a real orchestra, and they combine us only when you consider all of them together. And so this is the thing that we would like, that we are doing. We are trying to build on the very same optical table microscope, combining different mechanisms of interrogation of biological samples. Not only because we are from Genova and we want to buy only one laser instead of buying more lasers, one per microscope. Okay, not only for this. And this is what we have done, and we can do a lot of things. I will be back on this later. I told this story to Marta Fioravanti. She is not interested in science, and she decided to offer this graphic representation. So you have some ideas. You have your microscope, you have your sample, you have your visible light, and then he decided to suggest to insert here artificial intelligence. And we called these momics. But now there is one point when we deal with an optical microscope, any instrument, an optical microscope in this case, spatial resolution. And maybe you have in your mind the so-called Abbe law, that is this one. Please read this paper, because Abbe never wrote that formula. Abbe made an experiment using monochromatic light, dealing with the frequency domain, establishing some concepts that are in tune with the Abbe formula. But he never wrote in the German paper that everybody is citing the Abbe formula. You cannot find this. Huh? Not Abbe. That was before. However, this is an interesting paper by Colin Shepard about resolution and super resolution. Because now, when we use the term super resolution, what do you have in mind? What can you imagine, super? Well, this comes from a physicist that was Giuliano Toraldo di Francia. He used for the very first time the terms super resolution or super resolved. And the idea behind this was the time of the information science. So you had Shannon, you had Nyquist. The idea behind, and reported in this paper, is that when you form an image, when we use this idea for forming an image, we are limited by diffraction in terms of instrument, for sure. But this is not the limiting aspect. The limiting aspect comes from the amount of information we are able to introduce in the channel that allows to form an image of the object. Any additional information. This guy, Lucas, posed a question that is very simple. What would you sacrifice to get a better spatial resolution? It's like a song by Giovanotti years ago. What would you sacrifice? The answer is the field of view. For those of you that worked with atomic force microscopy, there is a very small tip. With a point-by-point interrogation, there is only one point interrogated by time. So you are sacrificing the field of view, space, and you get a better information. Adding information means that you can use computation. In Genova, we had Mario Bertero, and you can use information about using a model of your system. What does it happen when light interacts with your system? And then you can try to figure out which is the origin of the signal that you are collecting. And I have to say that many years ago, these guys using computation got a very high resolution that they called the super resolution in 3D, performing computation. And the most important information, additional information you have when you use computation, can you imagine which is the first additional information you have when you perform computation and when you go to solve your inverse problem? The information you have is that your solution has to be positive because you are dealing with intensities. Dot. And you immediately get an increase of resolution when you try to solve your problem. And here in Padua, again, there was Sarino, Rosario Rizzuto, and Tullio Pozzan working with Fogarty and Fay at that time and getting super resolved images of biological samples. We did the same in Genova, and people was dealing with these inverse problems for a long time. Considering the spatial domain, you have a signal that is changing in space, and you can apply Fourier transformation here, and you can describe what happens to your signal in terms of Fourier transform. The condition is that the phenomenon exists in nature. This was from the Bracer book in Fourier transformation. And so you have frequencies. High frequencies are rapid changes of the signal in space, spatial frequencies. Low are low. 0 is the total energy. And when you have a sample and you don't know which are the frequencies, the distribution of your objects, you know that your microscope applies a filter, your instrument applies a filter like this one, so it's cutting some frequencies. If you go to the inverse of this number, you have the upper limit again. There are different ways for establishing which is the spatial resolution of your instrument. You can use a line, and so you can say which are the shortest distances you are able to distinguish, or you can use a model of the microscope, or you can use other systems. What we suggest is to use this that is called Fouriering correlation. You simply need to collect 2 independent images of your sample, performing the Fourier transformation, evaluating the cross correlation, and finding here, when you have established the threshold of noise, the effective cutoff frequency. It's a comparatively simple way that tells you which is, in an objective way, the spatial resolution offered by your system. It's not something that you decide in which region, or so you can trust this. Adding information means also, for Mario Bertero and Roy Pike, performing this game. I shine light on my object. I collect the information in one-point detector. This is what we do. What about collecting this information in a camera? So more information from the very same object pointed by the beam scanning. And also Colin Shepard, they wrote some theoretical papers on this. And Jörg Enderle implemented this in what we call today image scanning microscopy. That is more or less this one. We shine light point by point, and then we have a detector made of 25 in this case, or for 25 in this case windows, very sensitive windows, and we collect 25 different views of our objects, who collect more information, and we can get a better resolution. It's comparatively simple, what we can do today with this kind of approach. But now I also want to tell you that you have a double increase of information. One increase of information comes from the fact that you have 25 detector elements. The other one, with respect to wide field microscopy, is that if you excite point by point, it's like watching this room in this way and then scanning. This means that I know from which position the information comes. This is an additional information. What is nice with this sensor is that we are also able to detect the time of arrival of photons in a window, and the jitter here is 100 picosecond. Fluorescence lifetime is around the nanosecond, so we can detect in their lifetime the arrival of photons from the sample. So we can also perform spectroscopy. And this is what we did. So you can play with an improvement in resolution and some spectroscopic information, molecule by molecule. Let me skip this. How to say we patented this, we sold this to Nikon, and you can read here that they use this, maybe it's better here, that they use this for the newest commercial Nikon instruments, that is this one and Spark. And we also decided to have a startup. And the name is Genoa Instrument for a very simple reason, because in Genoa, we have only one football team, so we wanted to have only one name reporting about this, that Genoa, and this is the advantage we can get. But in terms of super resolution, these three guys got the Nobel Prize with this motivation for the development of super resolved fluorescence microscopy. So this changes completely the way we attack with a microscope a sample. This means that these are the fluorescent molecules. This is what we can see using a conventional microscope. And this is what we can see when we use a super resolved microscope. So we can distinguish better position of fluorescent molecules. This is a nuclear pore, so we can see the components of the nuclear pore in a clear way. You can start thinking that you can rewrite what is reported in biological textbooks, because now you see where things really are. For example, in the case of chromatin and in the case of other components of the cell. And the idea behind is comparatively simple. We are not beating the diffraction. The diffraction limit is there. The term we used is circumventing. Simple, by precluding the simultaneous emission of adjacent, spectrally identical fluorophores. Simple. You can apply this idea for fluorescence and non-fluorescent, for absorption and non-absorption, scattering, non-scattering, spin up, spin down. With fluorescence, it's comparatively easy, because we have a photo-switchable fluorescent. And since I come from Genoa, well, the Columbus egg is something famous, and this was the sentence reported, because the community was working on this. The difference, gentlemen, is that you could have done it, but I did it. And this is what they did. We are with our orchestra again. We are with our molecules at an exhibition. And the two main ways for producing super resolution are the following. Which are the information we add? For Betzig and Mueller, the additional information is that we are building an image considering molecules that are at distances larger than the diffraction limit. Single molecules, this is the key word, single molecules. With Hell, the idea is that we are able to identify a region, this one, this green, in a very precise way in terms of space, from which the sample, the fluorescence comes. And the red region is the region where stimulated emission is induced. So the molecule is in the excited state. It normally will decay in the green, but you push the molecule to decay in the red. You don't read the red, and so you form your image with this additional information. I want to spend just 1 minute or 2 minutes about single molecules. Because when we say single molecule, it seems something normal. This is maybe for young postdoc or students. How can you decide, how can you be sure that you are dealing with single molecules? Because your detector does not know. One way, so your single molecules, if you watch them using the lens, you are diffraction limited, so this is what you get. When your distance is larger than the diffraction limit, you can recognize them as single or not. Single. One experiment is the following. You can use intensity, light emitted. You distribute them in a sparse way on a surface. You collect the intensity spot. You produce this analysis, and you find that there is an intensity 1, a multiple of this intensity. And so you can say that regions having these two are aggregates of 2 molecules, 3 molecules, 7 molecules, and so on. And so now you can say that if this spot is 1, here you have a single molecule, and you are dealing with a single molecule. Maybe this is not enough. You can use another characteristics, and this is the real world. I don't have these bands, I have these, and I have these families. But then there is another way. If I have a single molecule, there is a property of the fluorescent molecules that the fluorescent molecules share with you. You stop listening when it's too long. I'm sure I'm too long now. They stop fluorescing. The difference is that you can get a coffee and you can start again. They stop forever. So if you shine a light on a single molecule, you have a drop down like this one, if it is one molecule. If you have an aggregate of molecules, you don't have all of them sleeping at the very same time. You have one starting sleeping, the other one waking up, the other starting sleeping, and so on. And so you can distinguish in this way. If a cluster is a single molecule, this is the real situation. Or, more interestingly, you can use polarization. A single molecule is an oscillating dipole. You have your analyzer. You turn your polarizer having the maximum. And so you are aligned. If you move 90 degrees apart, if it's a single molecule, you have a 0 signal. If not, you don't have a single signal. If R2, it's difficult that they are oriented in the very same way. Only to tell you that when we talk about single molecules, our knowledge of the single molecule is this one. So we made a lot of experiments, and we do, while collecting the information, this kind of check, cross-check on the molecules. Betzig built his system at home. This was the home of a friend, Harald Hess, because Betzig's wife was not happy to have the microscope in the room at home, so he moved to another room. Harald Hess was not married at that time. He built the instrument in this way. He received some fluorescent molecule from a friend, and these molecules, the molecules he used, were photoactivatable molecules. So were molecules that were emitting 0 light. Then you can photoactivate them, they start emitting. If you photoactivate a very small number, you have a high probability that their distance is larger than the diffraction limit, and you continue this process of photoactivating, having photobleaching, and collecting them, summing them. This is a wide field image, and finding the position of the center of emission of each single molecule gives you a super resolved image, like in this case again. At the end, here you will find an image that you can say, I know this image, this is a cytoskeleton, right? But with this cytoskeleton, I know the position of each molecule with a precision of 15 nanometer. And I can play this game, just to continue with a single molecule, also with big objects. This is a tumor, a spheroid. These are the cells there. And in order to remove the background, we decided to shine light in a different way. Usually we excite light in a lazy way. So I shine light and I collect the signal. If I like to make gym, I don't, but in this case I did. I can shine light, I can move 90 degrees apart, and I can collect signal. So I remove all the background below and in front of the sample. And in this case, I can apply a single molecule approach in my big objects, that is the tumor spheroid, plane by plane, detecting the position of single molecules. Of course, I have some problems, and the problems I have are that when I deal with single molecule, this is the distribution of single molecule. This is a kind of perfect photoactivation and illumination of the single molecule, so I can form an image that is similar to the real distribution. In case is poor the localization or is poor the labeling, I get these two. In case molecules are moving, I am in troubles. I was in troubles. Because starting from this problem, the community started to find methods for tracking single molecules. The method is this one. This is your brush. You shrink the information using stimulated emission. So you play this shrinking and you get information only from this region. And so your brush is this one. And since you are working with a scanning system, you can decide in which region of the sample you can apply your improvement of resolution. And if this is the conventional image of the nuclear pores on the surface of a nucleus, this is what you can get using shrinking of the information collected point by point. The main difference between this method and the other one is that the other one requires a very large number of molecules to build an image and a lot of computation. In this case, you have the improvement in real time. Your ability of shrinking depends on the power, on the intensity of the second beam. There are a lot of problems in using this very high intensity, but if your second beam has a zero intensity, a zero power, you have the normal image. You can increase spatial resolution, you can improve spatial resolution. And if you use Fourier ring correlation, this is what you get. This is your cutoff frequency, low power. And this is your cutoff frequency when you increase the power. Increasing the power can be dangerous for the sample. We can discuss later if you want about this. And so we have to find a way for solving this point. But what are we doing with this microscope? We excite, we deplete using the stimulated emission, and we get this. So we perturb fluorescence here, but we said at the very beginning that when we perturb fluorescence, lifetime changes. So time of arrival of photons changes. This is the time of arrival of photons from the center, and for photons that are not killed in terms of green emission from the periphery. If you apply gating in your acquisition, you can select using time the position of the molecules that are delivering the signal. And so you can use time for decoding space. And this is what happens, for example, in the commercial microscope. This is the Leica 1. You have the confocal, you have STED, and you have, now you use the lifetime information, and you can improve your ability. And you can apply this in different ways and different conditions, removing background and having other advantages in using this additional information. I don't know if you know, the old one, yes. The young, less. Chang Lendl, Roland Garros. Huh? Chang did something completely unexpected and simple. Have you seen this match? This is what Lendl, this is Chang. Beating from the bottom. Roland Garros. Lendl became crazy. people watching, this never happened. He won the Roland Garros. A very simple idea from the scratch. And this is what Balzarotti had, dealing with single molecule. A very simple idea, like the one by Chang. And the method is called mean flux. You can have this, for example, this review in open access. The idea behind is the following. Until now, when I form my image, I use the maximum intensity I can collect from photons. When I use mean flux, in order to determine localization of the emitting molecules, I use the minimum number of photons coming from the region where the molecule is. This is the game for Betzing and Mueller, for L. This is the game of Betzing and Mueller, the single molecules, and then I build. In this way, I find a strategy. I still use a donut for exciting. I have the molecule somewhere. This emits photon, and then I move my donut with a certain strategy until I have the minimal number of photons collected. When I have the minimal number of photons, this means that the molecule, the single molecule, is at the center of the donut, because at the center of the donut, I do not excite. Okay? It's simple at the end. So this is what you do. This is your diffraction limited signal from that star. Your localization precision in conventional single molecule localization microscopy is dependent from the number of photons you are able to collect, square root of n. So here you have the upper limit, 200 nanometer, divided by the square root of the number of photons, 10,000. It's difficult, 10,000. However, this is the number. With the mean flux, you move your donut. You have to be sure that you are dealing with single molecules. You record the photon. At the end, the localization precision depends on the square root of n, the number of photons you collect. But is driven by your precision or ability in moving the beam. That is at least 2 orders of magnitude better than the diffraction limit. And so you need less photon. So when you play this game, and in the real world, what you do is that you start with a big donut, then you shrink around the single molecule. It's not so. It takes a long time, and this is the reason why we go to the artificial solon. Sorry. I try to speed up here. And so this is what you do. The stars are the molecules. You locate them one by one. And at the end, you have your view. And you can have your view in 3 dimensions. And these are the nuclear pores that are a gold standard for telling us if the system works. No, not in real time. In order to get this, it can take from 2 to 10 minutes, and in a very small region. This is the reason why we want to switch to the artificial. Now, the very first point was to play this game, I have to sacrifice space, the field of view. This means that I have to collect the information from a very small region. If I want to build a total image, I need really a long time. Is there any chance to understand in which regions I really want to get super resolution? And so we started working in transforming a conventional image in a mean flux image training, using the training of an artificial net. And so the point is this one. We have the confocal, we have the mean flux, we can collect them at the very same time, and we want to use these two images to train a net. And this is what we did. Alberto Morgantini, that was PhD in the Francesca Zanacchi lab, did this. Again, STED, single molecule, mean flux, and this reminder in terms of localization precision. Please note that here you have dependence from the wavelength and from the lens. Here you don't have. Is wavelength independent, lens independent. You start finding in papers these Ångström resolution fluorescence microscopy. You are able to localize room temperature, atmospheric pressure, position of fluorescent molecules, 5 Ångström localization precision. We are going to do nothing. We are simply, it's like like the fluorescent molecules, like the students, I don't know, both. Think that we are able to detect position of the fluorescent molecule that is linked to a biological object. So the idea now is that in this territory where there are a lot of molecules and things, and the molecules, and the fluorescent molecule, to be specific for a certain region of the cell are linked to an antibody, to use this antibody as a lazo is the constraint for a fluorescent molecule. Fluorescent molecule is able to move around and to tell about fluorescence properties what's going around in terms of viscosity, in terms of changes of pH, in terms of changes of other characteristics. In which region? Well, in a region that is comparatively small, 5 nanometer. That's nice. My comment is, I understand I'm very long, I have some other things, but think that we are in a new era of microscopy. Think that we are forming images dealing with the properties of single molecules, one by one, not cluster of molecules, increasing spatial resolution, nice things, but now things are changing. We are dealing with collection of information from single molecules. I will skip this. This is about the atomic force microscope. In case you're interested, you can ask later. Label-free, why? Shortly. You come to the university by bus. The bus is overcrowded. You know where the stop is and when you have to get out. And you organize yourself in the bus in a way to stop at the university, to get out. Are you sure that you can play the very same game if one day one comes to the bus and put on the shoulders of each one of you a backpack of the very same dimensions of you? Are you sure that you will organize in the very same way and that you can get down at the right stop? So when we use fluorescent molecules, the fluorescent molecules interact with the sample or go to the shoulders of your sample. They're big like the molecules we are interested in. And we have to add a mechanism of contrast. Now, if you go to the label-free, these are the few methods you can find, so we'll skip them. I just skipped some of these images. These are some intrinsic fluorescence, so it's label-free. This is second harmonic generation. Sorry for this, but I didn't want to discuss about this. I want to go to chromatin DNA, because my interest is understanding when this chain opens and releases the information. Then I go to the coffee machine. When you are at the coffee machine, if you insert the coin in this way, you can get a coffee, probably, if the machine is working. If you try to insert the coin 90 degrees apart, it's difficult to get a coffee. If you throw your coin to the coffee machine, you can get a coffee someday. And so our coffee machine is the chromatin DNA, or the chromatin DNA opens and closing. The idea comes from Ignacio Tinoco many years ago, and the method is called circular dichroism. We control polarization of light. We use right or left circularly polarized light. And when is right or when is left and makes chromatin DNA condensed or organized in a certain way, light is more or less scattered or absorbed. We are interested in scattered light, not in the absorbed light. Carlos Bustamante is a very good physicist, had his PhD thesis in 1981. And there were some applications on chromatin at this time in 1988. The reason was very simple. They were dealing with circular dichroism. Circular dichroism means that you have differences between right and left as function of differential absorption. They had signals outside the absorption bands. And so this signal could come only, they demonstrated, only from scattering. Well, we built an instrument for understanding this many years ago, and we can use a formalism that is called Mueller matrix formalism. Is a matrix made of 16 elements. You describe your polarization in the input by a vector of 4 position. You describe your output in terms of polarization in the vector of 4 position, and the link is a 4 by 4 matrix. Each of these elements tells you something about the sample, including birefringence, this one. This one is about chiral objects. To modulate light, comparatively simple, you can use a quartz, oscillating quartz, and changing polarization several times. And so you can have, from each of these elements, for free, without labeling, information about dichroism, the polarization, and so on, birefringence. This is the map you can build for a birefringent sample or for a chiral sample. We decided to attack with this system using a chiral an object on a helical trajectory that are sperm heads of an octopus. You can get this only in May. Then we had some model about the interaction of right and left circularly polarized light with objects organized with different condensation in helical structures. And then we decided to build our instrument. Our instrument is sensitive. This is what we are checking still now, in a precise way, to the chiral states or to the condensation or decondensation of chromatin using the Mueller matrix formalism and the Stokes vector input and output. System is comparatively easy, so you have a source of polarized light that you have to control. We bought on eBay for 300 euros this Zeeman laser that allows you to have the, by magnetic field, the change of polarization at a very high frequency. Or you can use a photoelastic modulator, but the core of this instrument is the control of polarization as input and output. And then we started collecting information coupled from circular intensity differential scattering and fluorescence. Fluorescence of what? Of DNA. When you see high intensity, chromatin is highly condensed. When you see low intensity, chromatin is less condensed at the very first glance. And so these are the images we can use now for training a system. I will skip this, but just to tell you that we have a lot of problems with the label-free signal, because in the label-free, when you are dealing with the label-free in this transmission issue or scattering, you have a bright background against a dark signal, and not a bright signal against a dark background. And so we decided to work in the dark field, improving the signal from the label-free, that is this one with respect to fluorescence. Sorry for this. And so for our molecule star exhibition, now that we are able to produce this couple of images, we have a more modern view. I don't know if you know Emerson, Lake and Palmer. Maybe you're too young, but maybe others, they know. And this is the extension of the Mussorgsky representation of the picture as an exhibition in progressive music. And the artificial microscope projects, again, want to, starting from a vision of the real world, to produce an artificial world that is reporting about the real one. Artificial intelligence. Artificial intelligence is old, 60 years at least. Nothing new. And from people that work on this, from the basic, you have unsupervised or supervised learning, but you mainly observe, and then you try to correlate things in different ways with good mathematics, but this is what you do. At the very beginning, we started using an upfield idea about recognizing patterns of chromatin using associative memories, just using artificial intelligence for a prediction of the healthy or unhealthy state of the cell. But more recently, the idea is to have this approach that is called paired image-to-image translation. The meaning is that I have two images. I train the system with two different images, and I want to have a system trained to produce the second one starting from the first. The first is label-free, and you can generate, starting from a label-free, a fluorescence image, and then you have a discriminator that tells you about what you are doing in terms of real or fake information you're building. We applied this for different couples of fluorescence and label-free images. And these are the very first reconstructions. So this is the label-free signal, and this is the fluorescence signal. We started using, instead of using Mueller matrix, we use phase contrast. That is a special part of the Mueller matrix at the end. And these are the results that want to transform label-free into fluorescence, generated fluorescence, and this is the true fluorescence. There are possibilities of improving these just with these two steps. One is a step that is improving the generated fluorescence. But what is behind are these two steps. The very first one I've shown you before, that was from confocal to super resolution. So from confocal to molecular distribution of biological objects. And the other one, the last, that is label-free into confocal. So this is the chain we are following. We can add different mechanisms of formation of images. And what we need from biology of medicine is a robust system for understanding if we are working well. So if we are able to recognize health from unhealthy. This is the reason why we use this system for the neuroblastoma. Coupling all the system, all the methods we know, and we are able to have using our microscope containing all the methods. And so the final step is this one, using artificial intelligence, transforming a black and white image in a molecular content image, which is the perspective. I'm really at the end. imagine that you have a medical doctor collecting a black and white image and can see in his or her glasses the molecular content to decide what to do in the next, for example, in an operative field or in other conditions, or studying the system of interest. I am indebted with this group. This is more or less the end of the story of this observation. So we started from the moon, but we still are in a spherical shaped object that is the cell. I really thank all of you for your patience, and I'm sorry I was so long, but I'm open for discussion in case you have time to do that. I hope so. Thank you very much. And you are more than welcome if you want to attend this conference in biological physics in Genova. For students, there are special rates. And so,
thank you very much for this inspiring talk. So we have time for a few questions. Okay.
Alberto, thank you for your, Mario Bortolozzi. Thank you for your great talk and amazing results in your research. I have a couple of questions about your mean flux technique. I was wondering why in the formula, the final formula which is independent from the lambda and the numerical aperture, why the numerical aperture is not comparing? Because I imagine that the donut that you generate is dependent on the numerical aperture of the objective. And the second question is about in the mean flux technique, is it necessary to apply a immunostaining method which helps achieving the best resolution? I mean, in general, you combine two antibodies, the primary and the secondary, but then you can even apply the fluorophore directly to the primary antibody, or even you can use a Fab fragment antibody to reduce the dimension of your antibody, no? And so, which improves the resolution. I would like to know at what level the mean flux obtains the best results in this kind of technique.
There are no, about this last part, there are no great differences among this kind of preparation. What matters is the ability of blink disappearing and appearing of your fluorescent molecule. This is what we are interested in. So you want the molecule out and in order to be able to localize this in the best way. This is the only issue that we have. Then the preparation of the sample depends on the question. So you can infer, but if you infer, then maybe you don't have the answer you wanted from your sample as the positioning of the fluorescent molecule. For the first part, that is about the diffraction limit, you're right. If I shine on a wall the donut, since I'm using a lens, I am diffraction limit. What I have to be sure that I have is a zero in the center. So what matters is having a zero in the center. This is the reason why you don't mind about the extension of the donut, or how large is the central part, or how the valley intensity is following a certain profile. You want to have a profile that has a zero at the end of this profile. And in this way, you are independent from the lens. And you can shrink or enlarge the donut. What matters is having a zero in the center. No, it's true. It's true that this is not always possible. So you have a dirty zero in the center, and you have some light in the valley, and so this reduces your ability in localizing with high precision the objects, because you don't have a minimal number of photons like in the other case. But the formula comes from the theoretical approach of this, and it's not true that is L divided by square root of n, is L divided n elevated k divided by 2, and you have to determine every time the key. So when key is 1, is the square root of n. And when you perform tracking, you have another additional parameter that you can add for improving the resolution, that is factor 4. So you can really, when you track, you are able to improve your localization precision of a factor 4. The problem is that it takes really a long time performing this kind of operation, because you have to select the single molecules. If you are not dealing with the single molecules, with a single molecule, the system realizes this and stops with some parameters that you can define. You can insert something in programming the experiment with the mean flux, commercial or not commercial, the sequence, the way you perform the tracking, and you recognize the single molecule. Also, recognizing single molecule is a key factor. This is the reason why I was telling you something about the ways for recognizing single molecules. So the two elements are single molecules and the 0 at the center of the donut. If your donut is dirty, you get lost. Of course, you calibrate the system. With gold particles, you have to see the 0 at the center. So the job can be, so maybe I have to stay here. So the job is long.
Thank you for this fascinating talk. I miss a time scale. In any case, you are building your image point by point. Each point takes some time, and the total time is so much. In this time, second, minute, whatever it is, I don't know, the molecule has not to move. Each point of the molecule should move less than a few nanometers. So how is this time scale?
That we track.
The molecule move usually. Maybe the velocity is larger than that.
No. So thank you very much for this. First of all, when we perform a mean flux, in this case, we work on a very small region. And we are able to track very well the position of the molecule in this small region. And so when we build the image, we can put them in the right position. When the sample is a living sample, is moving. When we have a fixed sample, is not moving, from my point of view, is less interesting. And the main application of mean flux is not for forming super resolved images, but for tracking single molecules. It takes too much time building an image, a precise image, if you want to form a big image from the single molecule. This is the reason why I was telling before, we have to start thinking that we are dealing with single molecules. You are right, they're moving, and we can track them.
How does it take to make an image of a molecule?
From 2 to 20 minutes for a very small region that is 1 by 1 micron. That's small in the cell. This is the reason why the prediction confocal mean flux can help to find a region where you can physically zoom, and not digitally, physically reduce your investigation to get precise information. But from my point of view, I like very much the images we are able to produce. But the more interesting application of mean flux is related to track the single molecules, and to take advantage of this tracking, because while tracking, and this is what we are not still doing, we are exploring an environment. You have a LEAD, or the molecule is alone, is free, but you are exploring an environment, and you can learn a lot about this environment from the photophysical properties of fluorescence, that are small changes in lifetime, small changes in wavelength emission, and polarization changes. Because when you have a rotational yes or not, this means that you have a viscosity that is less or more, and so on. So you can really, Question about preparation of the sample is a key question, because there is no an agreement on what you can do when you change your preparation, when you decide to bind one molecule, when you are using two antibodies instead of one. You don't know where you are placing your molecule. You have an antibody that is 10 nanometer, you are placing your molecule where? Exactly. So really, this is a key point. But this key point is more related to the question biologists have.
Yeah, so thank you very much for this inspiring talk. I was wondering, at some point you mentioned the increasing power, and I was just curious about that. Like, are we, do you mean that?
The stead power, you mean?
Yeah, like you said, like, if you can tell something more, because I was wondering if this is connected to the fact that you're dealing with biological samples in a way.
Right. So
thank you.
This is one of the central questions about STED. Stefanel presented his work in Genova 2003 for the very first time at Focus on Microscopy. And the first question he had from Fred Brackenhoff was, what about photobleaching and your interaction with the sample from the side of the donut? Because it's true, so you can have stimulated emission using any wavelength in the emission spectrum. What matters is that you can use this for bringing the molecule to the ground state. You try to be on the right side, so to the red, because you are far from excitation and you don't want to re-excite. This is one point. Okay. The other point is that if you want to shrink more, you have to increase the intensity. More photons, higher is the probability of performing stimulated emission, right? You are in the red. It's not true that in the red your sample does not absorb. It's true that absorption goes down, so cross-section is very poor. But it's not true, and since you are using a very large amount of photon, you have the chance of having absorption, and so emission again from the sample with two problems, two drawbacks. One is that you have a fluorescence signal from a region that you don't want having signal. The other one is that, and you are consuming also fluorescence in terms of photobleaching. The other one is that you can have an increase of free radicals, for example, in that region. Okay? And so you want to reduce this power. But when you reduce this power, now you have to imagine, you have to try to imagine, that you have fluorescence coming from the center. That is the one you want. And some green fluorescence, imagine this, passing through the donut. The difference between them is not the color, the wavelength, the energy. They are both green. The difference is lifetime. Because when coming from the center, lifetime is the lifetime of the molecule. When passing through the borders, there is another chance of going to the ground state. So lifetime is shorter. And so you shine light, you make something like this, like when you listen to a thunder. You start counting, and fluorescence ending too early comes from the border. Instead of removing this signal, you can use this signal. You can collect all the signal, and this is useful for removing background by subtraction, because in this case, background is uncorrelated with the signal you are collecting. It's the only case where you can remove background by subtraction without too many risks that usually you have when you remove a background by subtraction. And this is the story about intense power, more or less. And also there is the aspect of cost of the laser, come from Genova. No, but there is another one. In case you want to multiplex this action, so you want to parallelize this in order to be faster, you need a very big laser if you are not able to reduce the intensity. And you can play this game by getting
Thank you.
More questions? So, if not, let's thank again Professor
Grazie
Diaspro.
a voi. Spero che un caffè sia sufficiente per farvi riprendere. Hope that the coffee will be enough for restarting with life.